are warmly welcome to one more question and answer session with Daniel Hayford. Over the next few minutes, I would like to respond to the question, what are the major techniques employed in the field of arthropodology? Indeed, the field of arthropodology involves various techniques for collecting, identifying, studying, and analyzing arthropods. These techniques vary depending on the specific group of arthropods being studied, their habitats, and the research objectives. One of the major techniques used in the field of arthropodology is arthropod collection techniques. There are two kinds of arthropod collection techniques, namely the active collection techniques and the passive collection techniques. The active collection methods involve actively searching for and collecting arthropods. They involve the use of the strip netting technique, hand-picking technique, beating sheet method, and the use of aspirators or putters. The sweep netting technique involves the use of fine meshed nets. In this case, a fine meshed net is used to sweep through vegetation to collect flying or resting insects. It is commonly used for butterflies, grasshoppers, and other flying insects. In the case of the hand-picking technique, arthropods are manually collected from surfaces, under rocks, or in soil. This hand-picking method is useful for larger insects such as beetles and caterpillars. For the beating sheet method, a cloth or plastic sheet is placed under vegetation and branches are beaten to dislodge arthropods. This beating sheet technique is used for spiders, beetles, and caterpillars. The aspirators or putters are small, lightweight suction devices used to collect tiny insects. They are often used for small flies, including black flies, mosquitoes, ants, and aphids. The passive arthropod collection methods involve setting up traps to capture arthropods without direct human intervention. The passive arthropod collection methods involve setting up traps to capture arthropods without direct human intervention. They include pitfall traps, light traps, malaise traps, Bailey's funnel or tool green funnel, and sticky traps. For the pitfall traps, a cup or container is buried at ground level and filled with a preservative or bait. It is used for collecting ground-dwelling arthropods like beetles, ants, and spiders. In the case of the light traps, a light source, for example, UV light, attracts nocturnal insects such as moth. It is often used in biodiversity studies. The malaise traps is a tent-like structure that guides flying insects into a collection chamber. It is useful for collecting large numbers of flying insects like flies and wasps. In the case of the Bailey's funnel or the tool green funnel, a heat source dries out soil or leaf litter forcing small arthropods to move downward into the collecting container. It is used for soil-dwelling mites, springtails, and other micro-arthropods. In the case of the sticky traps, an adhesive coated surfaces capture flying or crawling arthropods. It is commonly used in pest management studies. Other major techniques used in the field of arthropodology are arthropod identification and taxonomic techniques. There are two types of identification and taxonomic techniques, namely morphological identification technique and molecular identification technique. In the case of the morphological identification, arthropods are identified based on their physical characteristics or external structures using dissecting microscopes. Compound microscopes are also used for the identification of smaller features like insect wings, legs, and mouth parts. The morphological identification employs taxonomic keys and guides for classification of arthropods. The molecular identification, which involves DNA barcoding and genetic analysis, involves DNA extraction and PCR or polymerase chain reaction. It is used to analyze genetic material for species identification. It also involves DNA barcoding, which uses specific gene sequences such as COI gene for insects to identify species. The molecular identification technique also involves next generation sequencing or NGS. The next generation sequencing or NGS allows for large-scale genetic analysis of arthropod communities. Other major techniques used in the field of arthropod studies are ecological and behavioral study techniques. Ecological and behavioral study techniques. These techniques include mark release rate capture (MRR) technique, radio telemetry and tracking technique, and high-speed video recording technique. Mark release rate capture technique, radio telemetry and tracking technique, and high-speed video recording technique. In the case of the mark release rate capture technique, arthropods are marked, released, and later recaptured to estimate population size and movement. This technique is used in ecological studies of butterflies, mosquitoes, and ants. For the radio telemetry and tracking technique, tiny radio transmitters are attached to larger arthropods to track their movement. It is used for studying beetles, bees, and other mobile arthropods. The high-speed video recording is used to study fast movement in arthropods, such as jumping in grasshoppers or predatory behavior in spiders. The fourth major techniques used in the field of arthropodology are experimental techniques such as behavioral assays, chemical analysis such as pheromone and pheromone studies, and toxicological studies. The behavioral assays involve observing arthropods in controlled environments to study their feeding, mating, and defense mechanisms. It is used in laboratory or field settings. The chemical analysis such as pheromone and pheromone studies are used to understand arthropod communication and chemical ecology. Gas chromatography mass spectrometry, or GCMS, is commonly used to analyze insect pheromones. The toxicological studies involve testing the effect of pesticides or biocontrol agents on arthropods. This kind of studies helps in integrated pest management, IPM. Imaging and microscopy techniques are also major techniques used in the field of arthropodology. These include scanning electron microscopy, SEM, confocal microscopy, and micro-CT scanning. The scanning electron microscopy provides high-resolution images of arthropods in terms of their structures. It is used to study microstructures like insect hair and compound eyes. The confocal microscopy uses lasers to produce 3D images of arthropod tissues and organs, whereas the micro-CT scanning provides 3D imaging of internal structures of arthropods without dissection. The last major techniques used in the field of arthropodology are statistical and computational techniques. The statistical and computational techniques include ecological modeling, geographic information system or GIS mapping, and machine learning and AI. In the case of the ecological modeling, software like R and Python is used for analyzing arthropod population trends and ecological interactions. The Geographic Information System or GIS mapping is used to track arthropod distributions, especially in disease vector studies, for example, mapping mosquito population. The machine learning and AI are used for automated identification of arthropods from images and ecological data analysis. The machine learning and AI are used for automated identification of arthropods from images and ecological data analysis. In conclusion, the field of arthropodology relies on diverse techniques ranging from traditional field collection methods to advanced molecular, imaging, and computational approaches. These methods help researchers study the diversity, behavior, ecology, genetics, and role of arthropods in different environments. Remember to subscribe, like, comment, and share with others in service to humanity. And see you in another question and answer time with Daniel Clifford.